Hi guys, welcome back to another tutorial video. In this video, we are going to talk about Gluconacetobacter diazotrophicus, considering as a potential biofertilizer which is widely used in the sugarcane field. So in this presentation, we are going to discuss about Gluconacetobacter, general characteristics, crop recommendation, isolation and identification, mass cultivation, and finally field application of Gluconacetobacter. In Graham's classification, under the microscopic observation, it shows pink color gram-negative rod with circular or rounded end, followed by it shows single, paired or chain structural arrangement. It was previously known as Saccharobacter nitrocaptans. However, it is renamed as Gluconacetobacter owing to withstand the acidic environment. It belongs to the family of Acetobacteriaceae. They are motile in nature due to the presence of flagella all over the surface, which is referred as peritrichous. Cap, endospore and cell inclusions are absent. In case of respiration, it is strictly aerob. Optimum temperature is 28 degrees Celsius and pH is 5.5. It shows and perform endophytic nitrogen fixation which is taking place in the stem of sugarcane. Originally, it was discovered by Valdemar and Johanna in the year 1988 in Brazil. For agriculture crops, the Gluconacetobacter is recommended to sugar excess plants such as sugarcane, coffee plant, pineapple, and finally sweet potato. Why? Because this bacteria is likely to grow especially in the sugar-rich crops. Next, how to isolate Gluconacetobacter from these crops. In nature, the Gluconacetobacter is found in phyllosphere as well as rhizosphere of the sugarcane. The root, shoot and sprouting buds were collected from these plants and washed with tap water and then sterilized. After the sterilization process, these samples were homogenized by means of mortar and pestle with 5% sucrose. Then, serial dilution were performed in 9 ml sterile water containing test tubes. This is how the isolation of Gluconacetobacter were done. Here, Gluconacetobacter microscopic characteristics were represented. Under the gram staining, it shows pink color gram-negative rod with circular or round-ended. Followed by it shows single, paired or chain structural arrangement. In hanging top method, as I told before, they are motile in nature due to the presence of flagella all over the surface, which is also referred as peritrichous. Cultural characteristics in the sense, here it is inoculated into 3 different media. Firstly, it is inoculated into semi-solid algae medium in which it shows orange color colonies with pellicle formation, followed by potato infusion agar which is supplemented with 10% sucrose solution or sugar in which it shows dark brown colonies, and finally GYC agar in which it shows brown color colonies. In biochemical test, under the enzymatic test, it shows positive for catalase and urease. For oxidase, it is negative. In vivo, it shows positive for indole and citrate utilization test alone. For protein and carbohydrate assay, such as gelatin, casein, and starch analysis, it shows negative. In nitrate reduction, it shows negative, and finally, it results positive for hydrogen sulfide and exopolysaccharide production. For mass cultivation of Gluconacetobacter, prepare LGA broth in Erlenmeyer flask and perform sterilization. Then mother culture was extracted from the target crops and incubated 3 to 4 days at 37 degrees Celsius. Then check out the purity of culture and then transfer to a large fermenter and wait for around 4 to 9 days for the visible bacterial growth. Once again, check the quality of broth after the fermentation and then mix the broth with desired carrier or liquid based material and then For carrier base, it is packed in polythene bags, and for liquid base, it is stored in bottles and kept in room temperature or 25 degrees Celsius. Then, how to apply the Gluconacetobacter for the sugarcane field? For that, after the 2 days of germination, 2 ml of liquid formulation of Gluconacetobacter was added to the soil around each seedling. At last, some interesting features of Gluconacetobacter were listed out. It helps to fix nitrogen in sugarcane tissue. It able to withstand the acidic environmental and pH. It also possesses osmotolerant up to 30%. It produces sucrose and also utilizes the sucrose sugar in different concentration. It forms pellicle and produce exopolysaccharide phytohormones such as indole acetic acid, and then significantly increase sugarcane yield. So, as far as we have discussed about the Gluconacetobacter as biofertilizer in various significant role in plays in sugarcane field. Thanks for your patient listening. Stay tuned, guys.